Let's ring the gong, yo. Whee! Okay, blog time, blog time. Whew. Oh man, this chapter is short. We'll probably get 2 or 3 done. Alright, next chapter. Hunting of the Sark. Alright, only quote. For the Snark was a boojum, you see? And that was from Lewis Carroll. Who knows what book? Alright, let's go. Sol Spiegelman had got hopelessly lost looking for cancer-causing retroviruses in humans. His predicament was symptomatic. Cancer biology, the NCI, and the Targeted Special Virus Cancer Program had all banked so ardently on the existence of human cancer retroviruses in the early 1970s that when the viruses failed to materialize, it was as if some essential part of their identity or imagination had been amputated. If human cancer retroviruses did not exist, then human cancers must be caused by some other mysterious mechanism. The pendulum, having swung sharply towards an infectious viral cause of cancer, swung just as sharply away. Temin, too, had dismissed the retroviruses as the causal agents for human cancer by the mid-1970s. His discovery of reverse transcription had certainly overturned the dogma of cellular biology, but it had not pushed the understanding of human carcinogenesis far. Viral genes could attach themselves to cellular genes, Temin knew, but this could not explain how viruses caused cancer. Faced with yet another discrepancy between theory and data, Temin proposed yet another bold conjecture, again standing on the thinnest foundation of evidence. Spiegelman and the retrovirus hunters, Temin argued, had conflated analogy with fact, confused messenger with message. Rous sarcoma virus could cause cancer by inserting a virus genes into cells. This proved that genetic alterations could cause cancer. But the genetic alteration, Temin proposed, need not originate in the virus. The virus had merely brought a message into a cell. To understand the genesis of cancer, it was that culprit message, not the messenger, that needed to be identified. Cancer virus hunters needed to return to their lamp-lit virus again, but this time with new questions. What was the viral gene that had unleashed pathological mitosis in cells, and how was that gene related to an internal mutation in the cell? In the 1970s, several laboratories began to hone in on that gene. Fortuitously, RSV possesses only 4 genes in its genome. In California, by then the hotbed of cancer virus research, the virologists Steve Martin, Peter Vogt, and Peter Duesberg made mutants of the Rous virus that replicated normally, but could no longer create tumors, suggesting that the tumor-causing gene had been disrupted. By analyzing the genes altered in these mutant viruses, these groups finally pinpointed RSV's cancer-causing ability to a single gene in the virus. The gene was called SRC, pronounced Sark, a diminutive of sarcoma. Sark, then, was the answer to Temin's puzzle, the cancer-causing message borne by Rous sarcoma virus. Vogt and Duesberg removed or inactivated Sark from the virus and demonstrated that the Sarkless virus could neither induce cell proliferation nor cause transformation. Sark, they speculated, was some sort of malformed gene acquired by RSV during its evolution and introduced into normal cells. It was termed an oncogene, a gene capable of causing cancer. A chance discovery in Ray Erickson's laboratory at the University of Colorado further elucidated Sark's function. Erickson had been a graduate student in Madison in the early 1960s when Temin had found retroviruses. Erickson had followed the discovery of the Sark gene in California and had been haunted by the function of Sark ever since. In 1977, working with Mark Collett and Joan Brugge, Erickson set out to decipher the function of Sark. Sark, Erickson discovered, was an unusual gene. It encoded a protein whose most prominent function was to modify other proteins by attaching a small chemical, a phosphate group, to these proteins. in essence, playing an elaborate game of molecular tag. In these, scientists had found a number of similar proteins in normal cells, enzymes that attached phosphate groups to other proteins. These enzymes were called the kinases, and they were soon found to behave as molecular master switches within a cell. The attachment of the phosphate group to a protein acted like an on switch, activating the protein's function. Often, a kinase turned on another kinase, which turned on another kinase, and so forth. The signal was amplified at each step of the chain reaction, until many such molecular switches were thrown into their on positions. The confluence of many such activated switches produced a powerful internal signal to a cell to change its state, moving, for instance, from a non-dividing to a dividing state. Src was a prototypical kinase, although a kinase on hyperdrive. The protein made by the viral Src gene was so potent and hyperactive that it phosphorylated anything and everything around it, including many crucial proteins in the cell. Src worked by unleashing an indiscriminate volley of phosphorylation, throwing dozens of molecular switches on. In Src's case, the activated series of proteins eventually impinged on proteins that controlled cell division. Src thus forcibly induced the cell to change its state from non-dividing to dividing, ultimately inducing accelerated mitosis, the hallmark of cancer. By the late 1970s, the combined efforts of biochemists and tumor virologists had produced a relatively simple view of Src's ability to transform cells. Root sarcoma virus caused cancer in chickens by introducing into cells a gene, Src, that encoded a hyperactive overexuberant kinase. This kinase turned on a cascade of cellular signals to divide relentlessly. All of this represented beautiful, careful, meticulously crafted work. But with no human cancer retroviruses in the study, none of this research seemed relevant immediately to human cancers. And that's our first break, and we'll mark it with Lil Bong. That wasn't good enough. There we go. Yet the indefatigable Temin still felt that the viral Src would solve the mystery of human cancers. In Temin's mind, there was one riddle yet to be solved: the evolutionary origin of the Src gene. How might a virus have acquired a gene with such potent, disturbing qualities? Was Src a viral kinase gone berserk, or was it a kinase that the virus had constructed out of bits of other genes, like a cobbled-together bomb? Evolution, Temin knew, could build new genes out of old genes. But where had RSV found the necessary components of a gene to make a chicken cell cancerous? At the University of San Francisco, or UCSF, in a building perched high on one of the city's hills, a virologist named J. Michael Bishop became preoccupied with the evolutionary origin of viral Src. Born in rural PA, where his father had been Lutheran minister, Bishop had studied history at Gettysburg College, then drastically altered his trajectory to attend Harvard Medical School. And after a residency at Massachusetts General Hospital, he had trained as a virologist. In the 1960s, Bishop had moved to UCSF to set up a lab to explore viruses. UCSF was then a little-known backwater medical school. Bishop's shared office occupied a sliver of space at the edge of the building, a room so cramped and narrow that his office mate had to stand up to let him through to his desk. In the summer of 1969, when a lanky, self-assured researcher from the NIH, Harold Varmus, then on a hiking trip in California, knocked on Bishop's office door to ask if he might join the lab to study retroviruses, there was hardly any standing room at all. Varmus had come to California seeking adventure. A former graduate student in literature, he had become enthralled by medicine, obtained his MD at Columbia University in New York, then learned virology at the NIH. Like Bishop, he was also an academic itinerant, wandering from medieval literature to medicine to virology. Lewis Carroll's Hunting of the Snark tells the story of a motley crew of hunters that launched an agonizing journey to trap a deranged, invisible creature called the Snark. The hunt goes awfully wrong. Unpromisingly, as Varmus and Bishop set off to understand the origins of the Sark gene in the early 1970s, other scientists nicknamed the project the Hunting of the Sark. And that's our second break, and we'll have another bong. Oh, that was a good one. Hell yeah. Let's go. Varmus and Bishop launched their hunt using a simple technique, a method invented in part by Sol Spiegelman in the 1960s. Their goal was to find cellular genes that were distantly similar to the viral Sark gene, and thus find Sark's evolutionary precursors. DNA molecules typically exist as paired complementary strands like yin and yang, that are stuck together by powerful molecular forces. Each strand, if separated, can thus stick to another strand that is complementary in structure. If one molecule of DNA is tagged with radioactivity, it will seek out its complementary molecule in a mixture and stick to it, thereby imparting radioactivity to the second molecule. The sticking ability can be measured by the amount of radioactivity. In the mid-1970s, Bishop and Varmus began to use the viral Sark gene to hunt for its homologs, using the sticking reaction. Sark was a viral gene, and they expected to find only fragments or pieces of Sark in normal cells, ancestors and distant relatives of this cancer-causing Sark gene. But the hunt soon took a mystifying turn. When Varmus and Bishop looked in normal cells, they did not find a genetic third or fifth cousin of Sark. They found a nearly identical version of viral Sark lodged firmly in the normal cell's genome. Varmus and Bishop, working with Deborah Spector and Dominique Stellin, probed more cells, and again the Sark gene appeared in them. In duck cells, quail cells, and geese cells, closely related homologs of the Sark gene were strewn all over the bird kingdom. Each time Varmus's team looked up and down an evolutionary branch, they found some variant of Sark staring back. Soon the UCSF group was racing through multiple species to look for homologs of Sark. They found Sark in the cells of pheasants, turkeys, mice, rabbits, and fish. Cells from a newborn emu at the Sacramento Zoo had Sark. So did sheep and cows. Most important, so did human cells. Sark, Varmus wrote in a letter in 1976, is everywhere. But the Sark gene that existed in normal cells was not identical to the viral Sark. When Hidesaburo Hanafusa, a Japanese virologist at Rockefeller University in New York, compared the viral Sark gene to the normal cellular Sark gene, he found a crucial difference in the genetic code between the two forms of Sark. Viral Sark carried mutations that dramatically affected its function. Viral Sark protein, as Erickson had found in Colorado, was a disturbed hyperactive kinase that relentlessly tagged proteins with phosphate groups and thus provided a perpetually blaring on signal for cell division. Cellular Sark protein possessed the same kinase activity, but it was far less hyperactive. In contrast to viral Sark, it was tightly regulated, turned on and turned off during cell division. The viral Sark protein, in contrast, was a permanently activated switch, an automaton, as Erickson described it, that had turned the cell into a dividing machine. Viral Sark, the cancer-causing gene, was cellular Sark on overdrive. A theory began to convulse out of these results, a theory so magnificent and powerful that it would perhaps explain decades of disparate observations in a single swoop. Perhaps Sark, the precursor to the cancer-causing gene, was endogenous to the cell. Perhaps viral Sark had evolved out of cellular Sark. Retrovirologists had long believed that the virus had introduced an activated Sark into normal cells to transform them into malignant cells. But the Sark gene had not originated in the virus. It had originated from a precursor gene that existed in a cell, in all cells. Cancer biology's decade-long hunt had started with a chicken and ended, metaphorically, in the egg, in a progenitor gene present in all human cells. Rhesus sarcomavirus, then, was the product of an incredible evolutionary accident. Retroviruses, Temin had known, shuttle constantly out of the cell's genome, RNA to DNA to RNA. During this cycling, they can pick up pieces of the cell's genes and carry them, like barnacles, from one cell to another. Rhesus sarcoma virus had likely picked up an activated SARC gene from a cancer cell and carried it into the viral genome, creating more cancer. The virus, in effect, was no more than an accidental courier for a gene that had originated in a cancer cell, a parasite parasitized by cancer. Rhesus had been wrong, but spectacularly wrong. Viruses did cause cancer, but they did so typically by tampering with genes that originate in cells. We only got like a page left of this chapter. But that was our last break, so another bong it is. Oh, that was not good. Yeah, there we go. Science is often described as an iterative and cumulative process, a puzzle solved piece by piece, with each piece contributing a few hazy pixels of a much larger picture. But the arrival of a truly powerful new theory in science often feels far from iterative. Rather than explain one observation or phenomenon in a single pixelated step, an entire field of observations suddenly seems to crystallize into a perfect whole. The effect is almost like watching a puzzle solve itself. Varmus and Bishop's experiments had precisely such a crystallizing, zippering effect on cancer genetics. The crucial implication of the Varmus and Bishop experiment was that a precursor of a cancer-causing gene, the proto-oncogene, as Bishop and Varmus called it, was a normal cellular gene. Mutations induced by chemicals or X-rays cause cancer not by inserting foreign genes into cells, but by activating such endogenous proto-oncogenes. Nature, Roos wrote in 1966, sometimes seemed possessed of a sardonic humor, and the final lesson of Roos's sarcoma virus had been its most sardonic by far. For nearly 6 decades, the Roos virus had seduced biologists, Spiegelman most sadly among them, down a false path. Yet the false path had ultimately circled back to the right destination, from viral SARC towards cellular SARC, and to the notion of internal proto-oncogenes sitting omnipresently in the normal cell's genome. In Lewis Carroll's poem, when the hunters finally capture the deceptive snark, it reveals itself not to be a foreign beast, but one of the human hunters sent to trap it. And so it had turned out with cancer. Cancer genes came from within the human genome. Indeed, the Greeks have been particularly prescient yet again in the use of their term onkos. Cancer was intrinsically loaded in our genome, awaiting activation. We were destined to carry this fatal burden in our genes, our own genetic onkos. Varmus and Bishop were awarded the Nobel Prize for the discovery of the cellular origin of retroviral oncogenes in 1989. At the banquet in Stockholm, Varmus, recalling his former life as a student of literature, read lines from the epic poem Beowulf, recapitulating the slaying of the dragon in that story. "We have not slain our enemy, the cancer cell, or figuratively torn the limbs from his body," Varmus said. "In all our adventures, we have only seen our monster more clearly, and described the scales and fangs in new ways that reveal a cancer cell to be, like Grendel, a distorted version of our normal selves." Interesting. I'm gonna need to read some Beowulf. That seems very interesting. Oh, I need some water. Give me a second. Oh, much better. Let's continue. Oh, right. We have a new chapter, and since we did the bongs for each of the section breaks, we might as well do it for for when we begin an entire chapter, shall we? Alright. Well, it's currently leaning against a bong instrument, so I'm gonna have to put you down and then bong it and then put you back up again. Bah. bong. What a great way to start a chapter. Alright. Next one. The wind in the trees. The fine, fine wind that takes its course through the chaos of the world, like a fine and exquisite chisel, a wedge blade inserted. And that is from D.H. Lawrence. Oh, man. The developments of the summer of 1976 drastically reorganized our universe of cancer biology, returning genes again to its center. Harold Varmus and Michael Bishop's proto-oncogene theory provided the first cogent and comprehensive theory of carcinogenesis. The theory explained how radiation, soot, and cigarette smoke, diverse and seemingly unrelated insults, could all initiate cancer by mutating and thus activating precursor oncogenes within the cell. The theory made sense of Bruce Ames' peculiar correlation between carcinogens and mutagens, chemicals that cause mutagens in DNA produce cancers because they alter cellular proto-oncogenes. The theory clarified why the same kind of cancer might arise in smokers and non-smokers, albeit at different rates. Both smokers and non-smokers have the same proto-oncogenes in their cells, but smokers develop cancer at a higher rate because carcinogens in tobacco increase the mutation rate of these genes. But what did human cancer genes look like? Tumor biologists had found Stark in viruses and then in cells, but surely other endogenous proto-oncogenes were strewn about in the human cellular genome. Genetics has two distinct ways to see genes. The first is structural. Genes can be envisioned as physical structures, pieces of DNA lined up along chromosomes, just as Morgan and Fleming had first envisioned them. The second is functional. Genes can be imagined, a la Mendel, as the inheritance of traits that move from one generation to the next. In the decades between 1970 and 1980, cancer genetics would begin to see cancer-causing genes in these two lights. Each distinct vision would enhance the mechanistic understanding of carcinogenesis, bringing the field closer and closer to an understanding of the core molecular aberration in human cancers. Structure, or anatomy, came first. In 1973, as Varmus and Bishop were launching their initial studies on SARC, a hematologist in Chicago, Janet Rowley, saw a human cancer gene in a physical form. Rowley's specialty was studying the staining patterns of chromosomes in cells in order to locate chromosomal abnormalities in cancer cells. Chromosome staining, the technique she had perfected, is much an art as a science. It is also an oddly anachronistic art, like painting with tempera in an age of digital prints. At the time when cancer genetics was zooming up to explore the world of RNA, tumor viruses, and oncogenes, Rowley was intent on dragging the discipline back to its roots. De Boveri and Fleming's chromosomes dyed in blue. Piling anachronism upon anachronism, the cancer she had chosen to study was chronic myelogenous leukemia, Bennett's infamous separation of blood. Rowley's study was built on prior work by a duo of pathologists from Philadelphia who had also studied CML. In the late 1950s, Peter Noel and David Hungerford had found an unusual chromosomal pattern in this form of leukemia. The cancer cells bore one consistently shortened chromosome. Human cells have 46 chromosomes, 23 matched pairs, one inherited from each patient. In CML cells, Noel found that one copy of the 22nd chromosome had its head lopped off. Noel called the abnormality the Philadelphia chromosome after the place of its discovery. But Noel and Hungerford could not understand where the decapitated chromosome had come from, or where its missing head had gone. Rowley, following this study, began to trace the headless chromosome in her CML cells. By laying out exquisitely stained photographs of CML chromosomes enlarged thousands of times, she typically spread them on her dining table and then leaned into the pictures, hunting for the missing pieces of the infamous Philadelphia chromosome. Rowley found a pattern. The missing head of chromosome 22 had attached itself elsewhere, to the tip of chromosome 9. and a piece of chromosome 9 had conversely attached itself.
Giano, what are you doing?
Attached itself to chromosome 22. This genetic event was termed translocation, the flip-flop transposition of two pieces of chromosomes. Rowley examined case after case of CML patients. In every single case, she found the same translocation in the cells. Chromosomal abnormalities in cancer cells had been known since the days of von Hansemann and Boveri. But Rowley's results argued a much more profound point. Cancer was not disorganized chromosomal chaos. It was organized chromosomal chaos. Specific and identical mutations existed on particular forms of cancer. Chromosomal translocations can create entirely new genes called chimeras by fusing two genes formerly located on two different chromosomes. The head of chromosome 9, say, fused with the tail of a gene in chromosome 13. The CML translocation, Rowley postulated, had created such a chimera. Rowley did not know the identity or function of this new chimeric monster, but she had demonstrated that a novel, unique genetic alteration, later found to be an oncogene, could exist in a human cancer cell, revealing itself purely by virtue of an aberrant chromosome structure. And that's a break, and that means we got a bong. Decent. In Houston, Alfred Knudsen, a Caltech-trained geneticist, also saw a human cancer-causing gene in the early 1970s, although in yet another distinct sense. Rowley had visualized cancer-causing genes by studying the physical structure of the cancer cell's chromosomes. Knudsen concentrated monastically on the function of a gene. Genes are units of inheritance. They shuttle properties or traits from one generation to the next. If genes cause cancer, Knudsen reasoned, then he might capture a pattern in the inheritance of cancer, much as Mendel had captured the idea of a gene by studying the inheritance of flower color on or plant height in peas. In 1969, Knudsen moved to the M.D. Anderson Cancer Center in Texas, where Freireich had set up a booming clinical center for childhood cancers. Knudsen needed a model cancer, a hereditary malignancy, whose underlying pattern of inheritance would reveal how cancer-causing genes worked. The natural choice was retinoblastoma, the odd rare variant of eye cancer that de Gouveia had identified in Brazil with its striking tendency to erupt in the same family across generations. Retinoblastoma is a particularly tragic form of cancer, not just because it assaults children, so, like every single modern billionaire, but because it assaults the quintessential organ of childhood. The tumor grows in the eye. Afflicted children are sometimes diagnosed when the world around them begins to blur and fade, but occasionally the cancer is incidentally found in a child's photograph when the eye, lit by a camera flash, grows eerily like cat's eyes in a lamplight, revealing the tumor buried behind the lens. Left untreated, the tumor will crawl backwards from the eye socket into the optic nerve and then climb back into the brain. The primary methods of treatment are to sear the tumor with high doses of gamma radiation or to enucleate the eye surgically, leaving behind an empty socket. Retinoblastoma has two distinct variants, an inherited familial form and a sporadic form. De Gouveia had identified the familial form. Children who suffer from this familial or inherited form may carry strong family histories of the disease. Fathers, mothers, cousins, siblings, and kindred affected, and they typically develop tumors in both eyes, as in de Gouveia's case from Rio. But the tumor also arises in children with no family history of the disease. Children with this sporadic form never carry a history in the family, and always have a tumor in only one eye. The pattern of inheritance intrigued Knudsen. He wondered whether he could discern a subtle difference in the development of cancer between the sporadic and the inherited versions using mathematical analysis. He performed the simplest of experiments. He grouped children with the sporadic form into one cohort, and children with the familial form in the second. And shifting through old hospital records, Knudsen tabulated the ages in which the diseases struck the two groups, then plotted them as two curves. Intriguingly, he found that the two cohorts developed the cancers at different velocities. In inherited retinoblastoma, cancer onset was rapid, with diagnosis typically 2 to 6 months after birth. Sporadic retinoblastoma typically appeared 2 to 4 years after birth. But why did the same disease move with different velocities in different children? Knudsen used the numbers and simple equations borrowed from physics and probability theory to model the development of the cancer in the two cohorts. He found that the data felt a simple model. In children with the inherited form of retinoblastoma, only one genetic change was required to develop the cancer. Children with the sporadic form required 2 genetic changes. This raised another puzzling question. Why was only one genetic change needed to unleash cancer in the familial case, while 2 changes were needed in the sporadic form? Knudsen perceived a simple, beautiful explanation. The number 2, he recalled, is the geneticist's favorite number, because they always have diarrhea. I'm kidding. Every normal human cell has 2 copies of each chromosome and thus 2 copies of every gene. Every normal cell must have 2 normal copies of the retinoblastoma gene, RB. To develop sporadic retinoblastoma, Knudsen postulated, both copies of the gene needed to be inactivated through a mutation in each copy of the RB gene. Hence, sporadic retinoblastoma develops at later ages because 2 independent mutations have to accumulate in the same cell. Children with the inherited form of retinoblastoma, in contrast, are born with a defective copy of RB. In their cells, one gene copy is already defective, and only a single additional genetic mutation is needed before the cell senses the change and begins to divide. These children are thus predisposed to the cancer, and they develop cancer faster, producing the rapid velocity tumors that Knudsen saw in his statistical charts. Knudsen called this the two-hit hypothesis of cancer. For certain cancer-causing genes, 2 mutational hits were needed to provoke cell division and thus produce cancer. Knudsen's 2-hit theory was a powerful explanation for the inheritance pattern of retinoblastoma, but at first glance it seemed at odds with the initial molecular understanding of cancer. The SRC gene, recall, requires a single activated copy to provoke uncontrolled cell division. Knudsen's gene required 2. Why was a single mutation in SRC sufficient to provoke cell division, while 2 were required for RB? The answer relies in the function of the 2 genes. SRC activates a function in cell division. The mutation in SRC, as Ray Erickson and Hidesaburo Hanafusa had discovered, created a cellular protein that is unable to extinguish its function. An insatiable, hyperactive kinase on overdrive that provokes perpetual cell division. Knudsen's gene, RB, performed the opposite function. It suppresses cell proliferation, and is thus it is the inactivation of such a gene by virtue of 2 hits that unleashes cell division. RB, then, is a cancer suppressor gene, the functional opposite of SRC, an anti-oncogene, as Knudsen called it. 2 classes of genes are apparently critical in the origin of cancers of children, he wrote. One class, that of oncogenes, acts by virtue of abnormal or elevated activity. The other class, that of anti-oncogenes, or tumor suppressors, is recessive in oncogenesis. Cancer results when both normal copies have been mutated or deleted. Some persons carry one such mutation in the germline, and are thus highly susceptible to tumor because only one somatic event is necessary. Some children, even though carrying no such mutation in the germline, can acquire tumor as a result of 2 somatic events. It was an exquisitely astute hypothesis spun remarkably out of statistical reasoning alone. Knudsen did not know the molecular identity of his phantasmic anti-oncogenes. He had never looked at a cancer cell to see these genes. He had never performed a biological experiment to pin down RB. Like Mendel, Knudsen knew his genes only in a statistical sense. He had inferred them, as he put it, as one might infer the wind from the movement of the trees. And that's our last break.